Hello, I'm Daniel Clark, a data science analyst in the Mayan Lab at the Icahn School of Medicine at Mount Sinai. Today I'll be talking about the gene set foundation model, a model we developed that was trained on over 1 million gene sets. In a nutshell, the gene set foundation model is a pre-trained denoising autoencoder using multi-hot encoded gene sets from these two resources, Remagene and Remagio. Remagene is a continuously updating resource of gene sets extracted from supplemental material of papers deposited in PubMed Central. And Remagio is a resource of differentially expressed gene sets from studies from the Gene Expression Omnibus. Both these databases were published in 2024, and GSFM is expected to come out in patterns this year. To train GSFM, we fed a binary vector encoding of each gene set through the network and try to predict the same vector by minimizing the binary cross entropy. At each stage, random dropout is applied, including on the input layer, forcing the network to learn redundant and high fidelity connections between partial subsets of genes. For evaluation, we randomly split the gene sets in half and see how well this and other state-of-the-art methods are able to recover the genes that weren't given to the model. Evaluation was done primarily on these four benchmarking libraries, and by comparing the distribution of area under the receiver operating characteristic curves produced by each method on gene sets in each of these libraries. GSFM-based methods significantly outperform or match all competitive methods on all the tasks we tried. Even GSFM trained on only gene sets published before 2015 or with additional synthetic noise added to the training data remained competitive when evaluated on these benchmarks. One key finding I'd also like to highlight is the saturation of method performance on the manually curated benchmarking libraries. Heavily literature biased methods like Tagger or ChatGPT in the gene PT similarity also perform well, but they do not perform well on data-driven benchmarks. Rumagene, however, does not exhibit this deficiency despite also being derived from literature. The autoencoder architecture is designed to be usable in separate parts. The encoder side takes a gene set and produces a dense gene set embedding vector with reduced dimensionality. The decoder side takes a gene set embedding and predicts how likely it is that any gene is in that set. A variety of tasks can be accomplished using the embeddings in much the same way the embeddings of large language models can be used, such as for gene set semantic search or for fine-tuning to new tasks. A future direction will seek to use these embeddings for large language model integration. Blitz GSEA is a fast implementation of GSEA developed by our lab. GSEA is usually performed by a weighted walk through a full gene expression signature. GSFM gene set enrichment analysis is achieved by using the GSFM assigned probabilities as weights for the same walk. In our benchmark, we used a dexamethasone gene expression signature and applied GSEA directly and compared it to the GSFM augmented GSEA based on only the significantly upregulated genes. We found that the GSFM augmented GSEA was as capable as GSEA on recovering the known target NR3C1. So this approach shows promise in simulating classic full signature enrichment analysis when only a set is available. Besides the libraries we used for benchmarking, we evaluated the final GSFM on many other gene set libraries and found, as expected, that GSFM is recovering missing genes from random splits of each set much better than random. We also evaluated GSFM on the full sets in these libraries to produce novel gene function predictions. The website we developed hosts gene pages for each gene, GSFM gene predictions, and an interface to use the GSFM model for gene set augmentation or enrichment analysis. As a case study for the GSFM website, we found this pathway figure from the Pathway Figure OCR project. It's a figure that describes the key pathways for autophagy and pancreatic cancer progression. We downloaded the human genes recognized in the pathway diagram and put them into the GSFM website augment feature. This evaluates the model using those genes and provides a table of ranked genes. We see on the first page many highly studied genes clearly involved in cancer pathways. Using GeneShot, our resource also developed in our lab that captures publications which mention the genes. We can get a sense of how widely studied each gene is in the GSFM prediction. On the right, we filtered the predicted genes by those mentioned by fewer than 50 publications. Despite being the 124th gene in the ranking, the first gene, ALDOA, can be found on PubMed as already being associated with both autophagy and pancreatic cancer. The gene INPP5K also has some evidence. In mouse experiments where it's been knocked out, it seems to lead to autophagy inhibition. And the Human Protein Atlas has marked it as a prognostic marker in pancreatic adenocarcinoma, among other cancers. RPS12, on the other hand, doesn't have much literature evidence for the connection. So if we plug it into the GSFM website, we can see a gene page that starts with the NCBI gene description, but also another tab called AI Overview. This description is made by providing GPT with the abstracts of up to 50 citations for the gene and asking it to summarize them and include citations. It definitely has its limitations, but can frequently provide more information than the, in many cases, outdated NCBI descriptions, especially with lesser studied genes. We see from the description that it's already implicated in tumor genesis and Wnt-dependent cancers are explicitly mentioned, a pathway known to play a role in pancreatic ductal adenocarcinoma. Also on the GSFM gene page are the pre-computed gene function predictions. CHIA 2022 predictions are transcription factors predicted to regulate the given gene. CAP1 is not yet known to regulate RPS12, but GSFM predicts that it might. CAP1 was already implicated in tumor genicity in pancreatic cancer, though, through regulating a different gene. More would need to be done to see if this gene plays a role or not, but hopefully we've illustrated one way in which GSFM can be used to find new directions. The next steps for this project include incorporating novelty on the results to better prioritize understudied predictions, adding more data and exploring larger models and different architectures to make even better predictions, and exploring other uses for the model, including integration with large language models. Again, the website we showed is public, and the model itself is available for use or repurposing on Hugging Face. With that, I'd like to acknowledge the members of the Mind Lab and thank the NIH for funding this research. And thank you for listening.